I'm Isabel Glazer,
I'm Katie Gettle.
and today we're talking about junk DNA. Okay, so basically, as you can see in the diagram, we have a series of genes, and in between the genes are DNA sequences that do not code for proteins. So, as I basically just explained, what is junk DNA? They're non-coding DNA sequences. So DNA, what is it? In general, it serves the role to code for certain proteins in the cell, but there are many different cells that not all genetic sequences in DNA could possibly code for all individual proteins. These DNA sequences typically come from a process called transposition, where there's a movement of sections of DNA to different positions in the genome. Most regions of junk DNA contain multiple transposons where sequences copy themselves and remove themselves from certain parts of the genome, and then they reinsert themselves in different parts of the DNA.
So the purpose of this junk DNA, a lot of it is used to make up non-coding RNA, such as transfer RNA, regulatory RNA, and ribosomal RNA. It also acts as a switch, which determine when and where genes get expressed. Other sequences are important for chromosome structure and the function of centromeres and play a role in meiosis. And some non-coding DNA sequences also determine the location where transcription factors can attach and control transcription of the genetic code from DNA to mRNA.
Okay, so now we're going to discuss some types of non-coding DNA, same junk DNA. Okay, so the first type of non-coding DNA is non-coding functional RNA, which is functioning RNA sequences that are not translated into proteins.
The second type is introns, which regulate tRNA and rRNA.
The third type is viral sequences, which are mutations that can deactivate in a viral genome.
And in class, we learned about telomeres, which are segments at the end of DNA that are used to prevent deterioration of the DNA during replication.
And we also have pseudo genes, which are related to known genes but can't code for a protein anymore.
And there's transposons, which may reverse a mutation or cause one.
And then we also have, lastly, cis and trans regulatory elements, which serve to be binded on and regulate genes. And then for a little fun fact about how much junk DNA we have, it's 98% of our DNA. So most of our DNA is in fact junk DNA.
Yes, and it's been around for hundreds of millions of years, so it shows that it was positive natural selection.
Thanks for watching.